Welcome to Cell Biology Unlocked, where we'll navigate the core principles that shape cellular life, from structure to systemic function. In this presentation, we'll introduce key concepts and the roadmap for the session, so you can connect molecular details to larger biological systems as we move through each topic together. This slide contrasts prokaryotic and eukaryotic cells and highlights the evolutionary leap of compartmentalization. Prokaryotes are small and lack a nucleus, while eukaryotes house complex, membrane-bound organelles. The diagram shows how gaining internal compartments enabled more sophisticated genome organization and paved the way for multicellular life. Here, we explore the fluid mosaic model, showing the phospholipid bilayer studded with integral proteins, peripheral proteins, carbohydrates, and cholesterol. The picture illustrates how this dynamic structure allows selective permeability, supports signaling and cell recognition, and maintains membrane fluidity, essential for homeostasis and communication with the environment. This slide breaks down membrane transport into energy-independent passive processes, like diffusion, facilitated diffusion and osmosis, and energy-dependent active transport, including pumps, and vesicular endos/exocytosis. The illustration clarifies how passive movement follows gradients, while active transport uses ATP to move substances against gradients or for bulk trafficking. Mitochondria are presented with their double membrane, cristae, and their own genome, reflecting an endosymbiotic origin. The slide highlights oxidative phosphorylation and ATP synthase as the engine of cellular energy production, and emphasizes ATP's central role, powering biosynthesis, transport, and mechanical work across the cell. This graphic traces protein synthesis from gene to destination, starting with transcription in the nucleus, mRNA translation at ribosomes, folding and quality control in the endoplasmic reticulum, and packaging in the Golgi for vesicular trafficking to the plasma membrane, lysosomes, or secretion, driven by signal peptides and chaperone guidance. Cell cycle regulation is summarized through G1 growth, S-phase DNA replication, G2 checkpoint, and repair, followed by M-phase mitosis, all coordinated by cyclin CDK complexes. The slide also shows outcomes when checkpoints fail, ranging from programmed cell death to uncontrolled division and oncogenic transformation, underscoring the importance of fidelity in replication. The mitotic process is laid out step by step. Prophase condenses chromosomes. Metaphase aligns them at the equator. Anaphase separates sister chromatids. And telophase reunites nuclei, while cytokinesis divides the cell. The visuals emphasize spindle microtubules and kinetochores as the mechanics ensuring equal chromosome distribution to daughter cells. Differential gene expression explains how identical genomes produce diverse cell types by modulating RNA polymerase access. Activators and enhancers promote transcription, while repressors and chromatin remodeling create silenced regions. The diagram connects these regulatory mechanisms to cellular specialization, showing how stem cells adopt neuron, muscle, or skin identities based on expression profiles. This slide describes post-transcriptional silencing by microRNAs and siRNAs, illustrating biogenesis steps, risk complex formation, and mechanisms of action. miRNAs typically fine-tune translation, while siRNAs can lead to precise mRNA degradation, and both pathways contribute to endogenous regulation and defense against viral or transposon-derived RNA. We survey advanced tools transforming cell biology, induced pluripotent stem cells for reprogramming and disease models, CRISPR for precise genome editing and functional genomics, and organoid cultures that recapitulate tissue architecture for drug testing. The slide also raises ethical questions about identity, germline editing, and equitable access to these technologies. Single-cell RNA sequencing is presented as a high-resolution approach that uncovers tissue heterogeneity, maps lineage decisions, and refines our understanding of disease mechanisms. The illustrations show how identifying distinct cell populations and developmental trajectories can inform cancer research, immune studies, and the push toward personalized medicine.